Good afternoon, everyone. I'm very happy to be virtually there or virtually here. And I would like also to thank very much Anne-Sophie and all the other organizers for inviting me to join this workshop, which I already attended a few years ago. And so I liked it very much. I'm very happy to be here. And while I share my screen, I would also like to thank John for the beautiful presentation that he gave, which really, I found it very interesting, and it also will help me a lot with my talk, because the basic physiological and biophysical matters were addressed very nicely. And so thank you, John, for your beautiful presentation. Can you see my slides?
Yes, we can.
Thank you. So I will start with this talk, which will be focused especially on EEG source analysis and on the mapping the brain activity and also the brain connectivity in the source domain, starting from EEG. So I prepared a short introduction on EEG origins and generation. Of course, John explained it very well, so I will be very fast on this. But I would like to start with the original view of EEG. If you are in the EEG field, you probably know Hans Berger, who was the first to find scalp potentials back in 1929, almost 100 years ago. And from the beginning, he expressed the hope that EEG would provide a window into the human mind. This was partially true from the beginning, because even in the earlier recordings, they were able to distinguish different mental states. So in some sort of way, it was true from the beginning. On another side, we are still struggling with the accurate localization of brain sources. So it's still not entirely true. We are working toward that. And I will show you a general description of the problems, even still open problems, and some applications. At the beginning of EEG history, we couldn't think, or they couldn't think of high-density EEG. In this picture, you can see the amplifier used to amplify a single EEG channel in 1934. So back then, it was impossible to think of increasing the spatial sampling of potentials over the head. But today, luckily, we are able to measure quite inexpensively many channels on the scalp, 64 and more, if we want to move to the so-called high-density EEG, which does not mean high spatial resolution still, but it's useful in that direction. And also the amplifiers are now very small and also portable. So this is something that we can deal with now. So the origins of EEG were already discussed very nicely by John. I will just shortly go to the dipole model. So the main today, it's we agree basically that the main contributors to the EEG scalp signals are the pyramidal neurons, which extend longitudinally in the cortex, and they have some properties which make them the best sources of scalp potentials that we can record non-invasively. Basically, their shape, which is, as I said, longitudinally distributed, their orientation with respect to the cortical surface. They're usually normally oriented with respect to the cortical surface, and their relative position, because they are disposed neatly in palisades, which means benches. So they are all in parallel disposition, which helps producing a signal that we are able to measure even at a distance from the sources, like we do with non-invasive measures. The single pyramidal neuron acts as a current dipole, but also groups of neurons act as current dipole. Starting from the single cell, just briefly recalling, we have internal currents, ion currents, and external changes of distribution of charges, which make the neuron act as an current dipole, which means that we have two charges, 2 point sources, positive and the negative ones separated by a distance. And this generates isopotential lines, which are the blue lines reported here, and return currents, which are the red lines here, which, thanks to the volume conduction, can be propagated across the tissues up to a certain distance. For the single neuron, this distance is not as large because the currents are quite tiny and quite small. But when neurons are put together and when they fulfill certain conditions, these currents can reach longer distances and even reach the scalp, where we are able to measure them. As I said, there are some conditions that must be met for this to happen. These conditions are that we need more neurons acting together in order to produce currents that are able to reach our sensors. So we need large groups of synchronous neurons. I will show you in a minute why it's important that they are synchronous, and also their disposition, the relative disposition is important. If these conditions are met, we can model by a dipole not just the single cell, but also groups of cells, even of many cells. The larger the group and the more met these conditions are, the stronger the signal that will be produced and that we will be able to measure. As I said, it's important how these neurons are located in the tissues. As I said, the main contributors to the EEG, to the scalp EEG, are the pyramidal neurons, which are longitudinally shaped and are also disposed in parallel in the cortex. However, generally speaking, this is not the only kind of source that can produce EEG. What's important is the kind of electrical field that is produced by the group of neurons. So if the neurons are organized in a way that produces a cancellation between the currents they produce in the tissues, we will have a closed field which we are not able to detect from a distance. So for instance, radially symmetrical neurons represent this problem of the cancellation of different effects. Also neurons which are longitudinally which have a longitudinal shape but which are randomly oriented can produce this cancellation, and even parallel neurons like the pyramidal ones can produce a closed field if they are asynchronously activated, because we have this cancellation effect between them. So what produces an open field is the summed effect of the synchronous activity of many neurons, which is met especially in the cortex, but also as the resulting activity of many neurons in other structures of the brain. And when this happens, then these regions contribute to the EEG, and so we are able, or we may be able to, we can be able to go back, to trace back this activity. Otherwise, that region is not visible to the EEG, and the EEG is blind to a region, means that we are not able to reconstruct it back. So it's important to keep this in mind when thinking of source models and what we are able to see and what we cannot see with a source analysis. Now, volume conduction, it's the heaven and hell of EEG scientists, because volume conduction is good, because otherwise we wouldn't have any EEG, any scalp EEG. So we are able to measure scalp potentials produced as a result of the brain activity because of volume conduction. At the same time, it generates a number of troubles. So we like it and we don't like it at the same time. Volume conduction, in brief, is the propagation of ion currents across the structures of the head, not just the brain, but all the structures of the head up to the sensors. As I said, this is the reason why we are able to measure something from outside of the head, but at the same time, it causes a number of problems or a number of limitations, which are well known when studying scalp EEG. So let's imagine that this is the brain. The blue dots are some sources in the brain. Some of them are cortical, and some of them are deep, are subcortical. These orange squares are the electrodes on the scalp. What happens when there's activity in one of these small regions of the cortex, for instance. Due to volume conduction, something happens here, and the currents are propagated in all directions, including these ones, and part of them reach the scalp surface. But there's a spread of these currents. There's also an attenuation and distortion, because the tissues are not, unfortunately, are not homogeneous nor isotropical. So there's a spread of the signal, and what we can measure on the scalp is much more spread, much more blurred than the original data, than the source of the activity. This is true for cortical regions. It's even more true for deep regions, because for a deeper region, the effect of volume conduction is worse from some respects, and deep regions can affect larger parts of the scalp, or even all the scalp. So there's this effect of spatial blur. There's a problem of attenuation of the signals due to the low conductance of some of these structures, which are interposed between the sources and the sensors, particularly the skull, as we have heard from the previous talk. And not only it's not only a problem of low conductivity, but also of anisotropy and of different layers between the tissues. And so the measures that we can get from the scalp are very prone to noise, and information is quite mixed up with a lot of noise. Another problem, which is important, especially for connectivity studies, which is something I'm particularly interested in, is the problem of the correlation of the signals on different scalp electrodes. Because given the not only we have the effect of a single source, which is spread on different electrodes, like here, but if we look at the signal collected on a single electrode, this will contain contributions from different sources. This source and this source, for instance, will both contribute to this electrode, as many others in the brain. So finally, when we collect and analyze the signal on a single electrode, it's difficult to refer this activity, to localize the sources of this activity. And if we look at close electrodes, the activity that we will record will be strongly correlated, not just or not only because the underlying brain activity can be synchronized or can be somehow interacting, but also because of this volume conduction effects. And this is not good, of course, because we want to study the true interaction between brain regions, and we need to get rid of this, or as much as possible to get rid of this correlation due to volume conduction and not to the interaction between regions. Finally, as I said, cortical sources produce very different contributions to EEG than deep sources, both because they are closer simply to the sensors, so they produce stronger and more focused kind of signals, but also because they usually produce an open field, while this is not always true, or it's usually not true for deeper sources. And as I said, when a source produces a closed field, EEG is blind to it, so we have a problem in localizing this activity, which is not contributing to the signal we are using. So we have a particular problem with deep sources. So these are the limitations. What can we do to improve things? What can we do to keep using EEG when we need to localize the sources? The first step, which was already introduced in the previous presentation, is to build a model, a physical and electrical model of the head, linking the activations at the brain level to the signals that are consequently produced on the scalp, and then we are able to measure. So the forward problem means computing what is the potential distribution on the scalp produced by a specific activation at the cortical or at the brain level. This is not a trivial problem. It requires a lot of information that we need to include in the problem. Basically, we need a model for the volume conduction, a model for the sources, and also a model for the sensors. As for the volume conduction, what we can do is to include information from different tissues. There are spherical models, there are more complex and realistic models obtained from MRI images. We can include information about the geometry and also about conductivity. If we want, and if we move to more complex models, we can also include anisotropies and inhomogeneity of the data into the model, with in particular with the FEM model. Moving from the simpler models to the more complex ones, there is no analytical solution. We need to move to a numerical solution. We need a source model. In the source model, we just need to put the dipoles that represent our sources, the number, position, and orientation of the dipoles depend on how coarse or how detailed our model is. It depends also on the geometrical information that we have. If we are using a spherical model, we will put the dipoles in the inner sphere. If we are using a realistic model, the dipoles can be positioned in the 3D space according to the geometry of the cortex or of the deeper regions. How many dipoles can we put in the model? Again, it depends on the information we have about the geometry and on how detailed we want our solution to be. Usually the range, the number of dipoles is around, it's between 3,000 and 10,000. These are typical values for these dipoles, which are distributed along the surface or along also the deeper regions. We can assign a fixed orientation according to anatomical constraints, for instance, normally oriented with respect to the cortex, or we can leave them the orientation free to be assigned by the method that we will use later. Finally, we need to put the sensors, the electrodes on the model. This is not a big problem. There are many solutions to this. However, we need to take care of that if we want to solve the problem correctly. Finally, what we do is to exploit the fact that the propagation from sources to sensors is described by Poisson's equation. Poisson's equation describes the propagation starting from the activation of a dipole and using the model previously described. The good point is that since the Poisson equation is linear, it obeys the superposition principle, which means that if we want to compute the effect of 3, for instance, sources simultaneously active to the scalp, we can obtain that effect by summing the separate effect of the first source by itself, the second and the third one, each of them considered as they were the only source active in that moment. So we can sum up the effects. We can use, turn on any single dipole in our model by putting its amplitude equal to 1, because since everything is linear, if I double the input, the output will be doubled as well. So if we put them to 1, we can build a matrix or a vector for each dipole of weights. These weights, keeping them all the information about the propagation from the sources to the sensors. The problem is not trivial. It's not easy to do this for a real situation, for a real condition. But finally, what we get is very powerful and simply to use, because we can build a matrix made of all these vectors, which is called the lead-feed matrix, which contains all the information about the geometry, the conductivity, the properties of the problem, and which is able to transform any given combination of source activations into the related scalp distribution, just by multiplying this matrix by the vector of sources. So the result is very, as I said, very powerful, very easy to use once we have it. Building it is not easy, but once we have it, it's very also fast to be used. However, there's a problem, there's an issue, which is not an issue for the forward problem, but will become an issue when we go back with the inverse problem, which is the fact that this matrix is strongly rectangular, because we have a number of sensors, which at best is of around 100, 200 electrodes, while the number of sources is much higher. This brings some problems when we want to go back to the sources with the inverse problem. In the inverse problem, we are closer to our real needs, because usually we don't measure the sources to obtain an estimation or an idea of what the scalp potentials would be, but we measure the scalp potentials and we need to infer which are the possible distributions that produce that scalp potentials. So this is a typical inverse problem in which we want to start from the data we record, which are the effects of some physical phenomenon, and we want to go back to the phenomenon that produced that result. As I said, we have a problem or a couple of problems. In fact, the inverse problem is strongly underdetermined and ill-posed. It's underdetermined because the number of variables is much higher than the number of sensors, as I just said, so there is no unique solution of this problem. There is no unique distribution in the source domain that can explain what we measure on the scalp. At the same time, and in addition to this, closed sources, which means different dipoles in the brain, usually generate very similar measures on the scalp, like in this example here. So the columns of the lead feed matrix are very close to each other, especially those related to closer dipoles, which makes the solution very subject to measurement errors. So it's a tricky problem. There were many different solutions that were provided to this, but what we need in any case is to add some constraints to the problem, because otherwise we won't have a unique solution. These constraints can be of different kinds, different origins. We can, for instance, include anatomical constraints. For instance, if we are aware of the structural connectivity linking some regions, if we're aware of the geometry of the head and of the head structures, we can include this information to improve the quality of the inverse problem solution. We can include the physiological constraints, for instance, the constraint of the correlation between close regions, which can be true in many conditions, but not always, but we can include this into the problem. Some biophysical constraints can be also included. For instance, the minimization of the energy of the sources is a biophysical constraint that we can introduce to reduce the number of solutions of the inverse problem. We can also include information obtained from different data acquisition methods, for instance, EEG or fMRI. These constraints can contribute to determine a unique solution of the inverse problem. Of course, the solution that we will obtain will be related to the assumptions that we made, and it will be as more accurate as these assumptions are verified by our data. So if our data do not verify the assumption, of course, this has an effect on the quality of the estimation. I would like to give you a short overview of the main methods and the main approaches that are used to solve this inverse problem. I will focus not on all possible methods because there are a lot of them, but I will try to give you an idea of some examples of different families of methods based on different approaches. And the first one, who was developed in the 90s, is the minimum norm solution, in which the constraint is that the sources have the minimum possible energy that allows them to solve the forward problem. So in the formula, we have a term which is related to the forward solution optimally explaining the data, this one. So this is the lead field matrix, this is the vector of the sources, this is the vector of the recorded potentials on the scalp. So we want to minimize this because we want the forward solution to be respected, but we also need the second constraint, which can be, like in this example, the the minimization of the least square error, the L2 norm, which means that we select the distribution of sources that minimizes the energy of the sources. So it's a balance between the forward problem and the minimization of the energy. There's also a regularization parameter that allows to obtain solutions which are physiologically plausible. There's a limitation in this approach, which is that given the requirement of the minimum energy of the sources, usually solutions are biased toward superficial sources, because a superficial distribution requires less energy to produce the same potentials on the scalp. So this is a bias of the method, because we know that actually there are deeper sources that contribute to the EEG. And to overcome this limitation, some weighting parameters were introduced to mitigate this bias, which led to the weighted minimum norm approaches. Among the weighted minimum norm approaches, one of the most commonly used is the so-called low-resolution electromagnetic tomography, LORETA, which adds an additional constraint, physiologically based, which is that the close regions are somehow related. There's a spatial coherence between them, which is obtained by minimizing the Laplacian, which means the second order spatial derivative of the current source distribution. This is a physiological plausible assumption when the dipoles are actually close on the in the brain. There are some conditions, for instance, around the sulci, in which this is not true. So the dipoles are close, physically close, but there's no reason to think that they should be correlated. So this is a strength, but also a limitation of the method. In particular, there were some improvements of the method to improve the localization accuracy, but they didn't affect the spatial resolution, which remains quite low. So we can use these methods if we want to localize the region, but we need to be aware that we will obtain necessarily a low spatial resolution over blurred solution. On the other hand, an advantage of this method is that it provides a distribution of the solutions in the whole brain volume, in the gray matter, but for the whole brain, and not just for the cortex, which can be very useful for some applications. So this is the first family of methods. During the decades, other approaches were developed. This is, of course, a non-exhaustive list, just few examples of the approaches that can be used. Some techniques are based on the spatio-temporal properties of the data. So again, a physiological constraint related to the variance of the data in the frequency domain and in the temporal domain. For instance, the beamformer uses a constraint about the variance of the dipole, of each dipole in time. So the limitation of this is again a smoothing issue and also a problem with correlated sources. So when we are dealing with correlated sources where we're particularly interested in the correlation between sources, this could be a problem. Other techniques are based on the scanning of all the possible locations of the source space to find out which is the most likely. So again, a different approach, different methods based on this. Again, there's a problem here, or there's a particular care must be taken when coherent and correlated sources are observed. Finally, there are more recently developed methods based on sparse theory. These are also very interesting. They are based on the assumption that in time, during short periods of time, only a limited number of cortical regions are actually active. So we can put some constraints on the dipoles that can be active and use a probabilistic inference. And these are also providing quite accurate results. So this is just to give you an idea of the variety of approaches that we can use, but they all have something in common, which is the information that we put into the problem. So we need the data collected on the scalp, but we need to feed our problem with as much information as we have about the physiology, about the anatomy, about the electrical properties, and about the behavior of the brain. Whatever we know, we put it into the problem. It will improve the solution, even if there is no method which is necessarily the best performing one. Before I move to a few final considerations on this, I would like to leave you here again a non-exhaustive list of all the academic, not all, but some academic and commercial software packages that are available to compute EEG source analysis. Some of them are academic and free, others are commercial packages, and they allow you. So if you're interested in one or more of these methods, you would find plenty of softwares that are able to do that for you. But there are some caveats. This is a quote that was already, heard today. I like it very, very much as well. All models are wrong. Some of them are useful. I put it here to remember that no matter how sophisticated the assumptions we make, no matter how informed are the constraints that we use, what we are using here is still a model, which means that it provides an estimation of what we really want to know, that is prone to errors, which is prone to, for instance, mistakes, also due to the fact that we need to be careful about the assumptions that we make. So a method can work perfectly fine when the assumptions that we make are satisfied by our data, but the same method used with a different data set, used with a different problem, could not be so good. And on the contrary, a different method could be preferable when the conditions are different. So what's crucial is to have a good knowledge of the properties of the method that we are using, and more importantly, of the properties of the data we are using, we are studying, and then we need to find a match between the methods and the data. There's no method that can be used anytime or that outperforms all the others in any condition. So this is a message that I found important, and I want to share it with you. In this last part of my talk, I would like to focus on connectivity studies because that's my main field of interest, and because connectivity is strongly affected by volume conduction and by the problems of scalp EEG. So I found it quite important to consider the implication of the source analysis for connectivity studies. You may have heard about brain connectivity, so this is just to introduce the topic. We know that neural populations are physically connected and are also functionally connected because they interact all the time during the brain functioning. So it's useful to study the brain functioning in terms of brain networks, both physical and functional networks, and the last ones can change in time with a temporal resolution, which is the same of the brain activity, so the millisecond scale. There are many possible definitions of interaction between brain regions. We can talk about synchronicity, coherence, or we can try to define a causality. Again, there are many different possible definitions of causality. One which is particularly useful and easy to use in neuroscience is based on temporal precedence. The main advantage of this definition is that it does not require an intervention to the system, so we don't need to perturb the system, we don't need to act on the system like other definitions of causality do. We just look at the data, look at the system, and search for the temporal precedence between brain activations according to the basic assumption that causes must precede their consequences. And so if we can detect this precedence, we can infer that might be some kind of causality between the brain regions. In particular, this is tested or can be tested by looking for any improvement in the predictive capacity between neural events. What does this mean? Let's imagine to have two time series. This definition was introduced by Wiener in 1956, and later Granger added a specific model, which is a very simple one, a linear autoregressive model. And according to this definition, if we can improve the prediction about the current sample of a time series by introducing information about the past of another time series, since the past helps predicting the future, we can infer that there's some kind of causality between these two activities, which is causality not in the physical way, but more in the statistical sense. So this is a definition which is quite simple, quite powerful, quite easy to apply to, in particular to EEG data. So it was very much used in the last decades, I would say. There is also a frequency version of that, which is based on a frequency version of the model, and which describes how causality in the statistical sense is distributed at different frequencies and allows to build a connectivity pattern like this at a specific frequency or at a specific frequency band. Again, there are many methods that are derived from this definition. We have the Granger test in the time domain. We have the linear, nonlinear, parametric, non-parametric versions. In the frequency domain, we have many estimators based on this basic assumption. So I won't go into details, but the general idea is that we are looking for the temporal precedence between the signals. And so the way in which they are correlated affects this estimation. So let's go back to the volume conduction. Are there any problems with that? Unfortunately, yes, because what we would like to occur is something like this: different sources, different activities on the scalp. If there is correlation, this correlation is due to a correlation between the sources. Unfortunately, this is not happening, or this is not the only thing that's happening. So there is a common effect of a single source on different electrodes. There are also there's also correlation due to currents moving through the different layers. So we have a problem when analyzing connectivity on the scalp level. That's why I will show now to you some examples of applications, sometimes also comparing what we can get from the scalp connectivity analysis and what we can get from the source domain connectivity analysis. Usual applications of source analysis are more in the field of epilepsy. I will show you something a little bit different, more related to basically three fields. The first one is the motor function, especially related to brain-computer interfaces application. The second one are cognitive functions, and the third one is the multiple subject analysis. So very briefly, in brain-computer interfaces, we aim to decode the brain activity intentionally produced by a subject to use this information for many reasons. First of all, or classically, historically, to create an alternate way of communication with the outside world for non-communicative patients. More recently, to create a different way to directly interact with the brain, for instance, for neurorehabilitation. Here, once we have solved the forward problem, as I said, and once we have solved the inverse problem, this is not simple, but as I said, finally what we get is a matrix which is very easy and fast to use, because we just need to multiply that matrix by our data, and we can get an estimation of what happens in specific sources. So this can be used even in real time to project or to infer the localization of the specific activity, with the advantage of have a higher capability of discriminate different parts, for instance, of the motor cortex, which can be extremely useful for brain-computer interfaces applications. Since I'm running out of time, I will be quite fast on these applications, but if you have questions, of course, we will have time later after after all the presentations. We also applied in a recent study, this was published last year, connectivity analysis in the source domain to identify different subtypes of psychiatric disorders, with very good results in the capability to discriminate between two different subtypes of post-traumatic stress disorder, and also measure depressive disorder, all based on source domain networks obtained from EEG at rest. So this is very easy to collect from patients, and this is a very important application. I think I will skip this because I have a few minutes and I have other applications which I would like to focus in. This one I would like to comment because it's based on the fusion, or better, of the inclusion of constraint derived from functional magnetic resonance imaging data into the EEG source analysis. So we can merge, or better, we can use the spatial information derived from, let's call it static, because of the low spatial temporal resolution fMRI data, into the solution of the source analysis, into the solution of the linear inverse problem, and this can improve the spatial resolution of our solutions by using these physiologically informed constraints. In this example, we obtain networks at the cortical level based on a minimum norm approach. The data were related to a cognitive task, which is the Stroop test. Finally, and last application I would like to show you is related to multiple subjects modeling. In multiple subjects modeling, what we do is to measure different subjects simultaneously while they interact. And the idea is to describe a complex system which is made of different individuals interacting and different brains mutually influencing each other. In this study, based on a social cooperation / defection task, which is called the prisoner's dilemma, we performed a source domain multiple subject connectivity analysis, returning a strongly characterized multiple subject network, able to distinguish, to classify the cooperative behavior from the defective one. So we classify this data with a high classification accuracy, and by the source analysis, we were able to look at the specific brain regions which were involved in this, which were in accordance with what we know about the social cognitive tasks, and also the involvement or not of specific networks during the different aspects of the task was unveiled by this source analysis. We did the same with other social tasks. I would like to skip to this last application, which is something we are doing now based on empathy and emotional alignment between different subjects. Here, I would like to show you a comparison of what we can get in terms of synchronization between the brains of two people involved in a task that elicits empathy. So one person is the subject of some bad or good treatment, the other is just an observer. There is an alignment between their brain activity that can be investigated at the scalp level. It's not impossible to see anything at the scalp level. Here, for instance, we see a synchronization between the two brains' activity. Of course, there's a lot of statistics behind to get rid of any spurious correlation. What we get is some kind of specific synchronization in time and located somewhere in the scalp. But if we move to the source domain, it's much more interesting what we can see, because here we can see the involvement of specific regions, both cortical and subcortical regions. This was made by Loretta. So the spatial resolution is not high, but the identification of key regions for empathy and for social interaction is quite accurate. And also the synchronization between the two subjects can be much better understood here with respect to what we can gather from the scalp. So with these few examples, I hope, my point is that it's not useless to do analysis in the scalp domain, but if we go to the source domain, we can infer much more interesting things. Of course, there's a lot of people I need to acknowledge, all my group, and you all for your kind attention.
Thank you very much, Laura, for this very nice talk.